Have you ever wondered what makes cancer cells different from normal cells? Today, we're going to take a deeper look into the complex world of cancer cells and their unique morphology. Cancer cells have some distinct features that set them apart from their healthy counterparts. Perhaps the most noticeable of these is their large nucleus, an organelle that acts as the control center of the cell. But it's not just the size that's different, the shape is too. Unlike the smooth, round nucleus of a healthy cell, a cancer cell's nucleus is irregular and often has prominent structures called nucleoli. These nucleoli are involved in the production of ribosomes, the cell's protein factories, which are crucial for the rapid growth and division of cancer cells. But the differences don't stop there. If we take a closer look, we can see changes in the chromatin, the substance within the nucleus that contains our DNA. In a cancer cell, there's a reduction in heterochromatin, a tightly packed form of DNA, and an increase in the number of nuclear membrane pores. These changes may contribute to the uncontrolled growth and division of cancer cells. Cancer cells also exhibit some interesting ultrastructural changes. The nucleus can have segments or invaginations, which are essentially folds or indentations in its surface. In addition, some cancer cells form inclusions within their nucleus. These inclusions are abnormal structures that can contain various substances, from proteins to viruses. These nuclear characteristics are just the tip of the iceberg when it comes to the unique features of cancer cells. Each of these adaptations allows the cancer cell to survive, grow, and divide, ultimately contributing to the development and progression of cancer. As we continue our exploration, we'll see that these nuclear changes are just one aspect of the remarkable transformation that a cell undergoes when it becomes cancerous. There are also significant changes occurring in the cytoplasm, the organelles, the cell membrane, and even the cell's metabolism. Now that we've understood the nuclear morphology, let's delve deeper into the other characteristics that make cancer cells unique. Did you know that cancer cells have an increased number of mitoses with atypical forms? Now, let's delve deeper into this fascinating aspect of cancer cell morphology. One of the unique characteristics of cancer cells is their atypical mitotic forms. Unlike healthy cells, which have a well-orchestrated and symmetrical mitotic process, cancer cells display a chaotic and asymmetrical style of cell division. This irregularity is often due to defects in the mitotic spindle, the structure that separates the chromosomes during cell division. In cancer cells, this spindle can be flawed, leading to an uneven distribution of chromosomes between the two new cells. Furthermore, cancer cells may exhibit dissymmetrical structures during mitosis, another divergence from the norm of symmetry seen in healthy cells. These unusual characteristics are not just random anomalies, they play a critical role in the uncontrolled proliferation of cancer cells, contributing to their rapid and invasive growth. These unusual mitotic characteristics are a key aspect of cancer cell morphology. Another striking feature of cancer cells is the changes in their cytoplasm and organelles. Let's delve into the cytoplasm first. Usually abundant in healthy cells, the cytoplasm in cancer cells is rather scarce, but it's not just about quantity. Quality matters too. In these cells, the cytoplasm undergoes notable transformations, including the appearance of new structures. It also takes on a basophilic hue due to the accumulation of ribosomal and messenger RNA. Intriguing, isn't it? Now, let's turn our attention to the organelles within these cells. The Golgi apparatus, often referred to as the cell's post office, is poorly developed in cancer cells. This might not sound like much, but the Golgi apparatus plays a critical role in sorting and modifying proteins, so its underdevelopment can have serious implications. Next up, the mitochondria. These powerhouses of the cell, as they're often called, see a reduction in volume as the tumor develops. This is because cancer cells have an altered metabolic state, relying more on glycolysis, a process that doesn't require mitochondria for energy production. But the changes don't stop there. The endoplasmic reticulum, or ER, is a network of tubules involved in protein and lipid synthesis. In cancer cells, the ER is simplified and frequently fragmented and degranulated. Both the granular and agranular endoplasmic reticulum undergo changes in amount and structure. So, just to recap, we've got a scarcity of cytoplasm, a poorly developed Golgi apparatus, a reduction in the volume of mitochondria, and changes in the endoplasmic reticulum. Each of these changes, alone or in combination, can significantly alter the function and behavior of a cell. And there you have it. From cytoplasm to organelles, cancer cells are a fascinating study in how changes at the microscopic level can have macroscopic effects. It's these cytoplasmic and organelle changes that significantly contribute to the malignant nature of cancer cells. Let's discuss the changes in the cytoskeleton, cell membrane, and metabolic processes of cancer cells. Our journey takes us first to the cytoskeleton. This intricate network of filaments and tubules gives cells their shape and aids in their movement. In cancer cells, however, the cytoskeleton undergoes significant alteration. We see different proportions of microfilaments, intermediate filaments, and microtubules, all critical components of the cytoskeleton. These shifts in composition and arrangement can profoundly impact the cell's ability to move and invade surrounding tissues, a hallmark of cancer's destructive nature. Next, we move to the cell membrane, the gatekeeper of the cell. In cancer cells, this barrier also undergoes drastic changes that influence tumor evolution and our body's responses. The cell membrane in cancer cells often displays modified surface receptors, new molecules, and abnormal antigens, all of which can trigger an immune response. Additionally, the cell surface reveals atypical structures teeming with highly active enzymes, contributing further to the malignant characteristics of cancer cells. Our final stop is the metabolic processes of cancer cells. Unlike healthy cells that efficiently use oxygen for energy, cancer cells show a preference for glucose, behaving much like metabolic parasites. This shift to a high glucose diet, even in the presence of oxygen, is a phenomenon known as the Warburg effect. Abnormal glycolysis processes occur in the mitochondrial membranes, further highlighting the metabolic quirks of cancer cells. These changes, whether in the cytoskeleton, cell membrane, or metabolic processes, are not random. They form part of a complex strategy that allows cancer cells to survive, thrive, and spread, often at the expense of the host organism. Understanding these changes provides invaluable insights into how we can combat this relentless adversary. These changes are fundamental to understanding the complex nature of cancer cells and how they operate.